For the past century, medicine has indexed biological health to a single variable, the count of years elapsed since birth. Standard screening protocols, mortality risk tables, and clinical treatment guidelines are all built on one embedded assumption: that the date on your birth certificate is the most useful available measure of what is actually happening inside your tissues. Yet the peer-reviewed literature from the past decade has produced a finding that challenges the foundation of that assumption. What your physician calls aging, and what every standard risk assessment measures with a birth date, is, according to the published biogerontological literature, a biological accounting error. The actual molecular state of your tissues can diverge from your chronological age by measurable years, and in several large prospective cohort studies, that divergence predicted all-cause mortality more strongly than the standard aging markers your physician currently orders, including measurements that have been in clinical research use for decades. This briefing examines the molecular mechanism responsible for that finding, the mortality data attached to it, and the controlled human trial evidence suggesting the clock running that mechanism has been run backward. The topic is the epigenetic clock. Specifically, what it measures inside your cells at the molecular level, what the peer-reviewed mortality data reveals about the cost of biological age diverging from chronological age, and what a series of controlled intervention trials found when researchers attempted to move that measurement in the opposite direction. The mainstream clinical model holds that aging is a continuous, broadly uniform process most accurately indexed by chronological age. Under this framework, a 65-year-old is assessed against standardized physiological norms for other 65-year-olds. The embedded assumption is that time elapsed is a reasonable proxy for biological state. What the research establishes is something fundamentally different. Two individuals sharing identical chronological age can differ in biological age by more than a decade. That gap is not a theoretical construct or a conceptual metaphor. It is a measurable molecular reality, captured from a blood sample using a technology that, in multiple large-scale prospective studies, outperformed several conventional markers in predicting who would die and when. This briefing will move through three mechanisms. First, the molecular architecture of biological aging and the mortality signal embedded in its measurement. Second, the discovery that biological aging has a real-time rate that diverges dramatically between individuals. And third, the trial evidence that this rate has been measurably altered in living human subjects. The findings presented here are drawn from multi-cohort prospective studies published in Genome Biology and Aging, involving validation populations exceeding 11,000 subjects, alongside a controlled human intervention trial with biological age outcomes tracked at 18 months post-intervention. The mechanism requires understanding one foundational concept about how your biology is actually controlled day to day. Think of your DNA as the hardware of a computer. It is fixed. It does not change. Every cell in your body carries essentially the same genetic code you were born with. But running on top of that hardware is an operating system, a separate layer that determines which parts of that code are active in any given cell at any given moment. Some programs are running, others are suppressed. The biological process that manages this switching is called DNA methylation. Here is how that switching system operates in practice. Across every cell in your body, your genome contains millions of specific positions that function like programmable switches on an industrial control panel. Each switch can be set to one of two states, active or silent. Small chemical tags produced by your body physically attach to these positions and lock the switch in place. When the tag is present, the corresponding genetic program is turned off. When it is absent, the program runs. In the clinical literature, these chemical tags are called methyl groups, and the specific positions on the genome where they attach are called CpG sites. In a young, healthy organism, this switching architecture is precisely calibrated. The right programs are running in the right cells. As an engineering system, it is operating within specification. Then time and environmental exposure begin to accumulate, and this is where the biological debt starts to build. The switching architecture does not stay calibrated indefinitely. Tags that should remain attached drift away from their positions. Tags that should be absent begin accumulating where they do not belong. Over time, the entire control system begins operating outside its original specification. Biological programs that should be running go quiet. Programs that should be suppressed become active. This progressive deterioration of the methylation switching architecture is what the clinical literature calls epigenetic drift, and it is not random noise. It follows patterns that prove to be remarkably consistent across individuals, across tissues, and across populations. In 2013, biostatistician Steve Horvath, working as a sole author, published a finding in Genome Biology that the field did not anticipate. Analyzing 8,000 samples drawn from 51 different tissues and cell types, Horvath identified 353 specific switch positions whose combined methylation state predicted chronological age with a Pearson correlation of 0.97. The median prediction error was 2.9 years, and the same 353 positions, using the same mathematical constants, predicted age with equal accuracy whether applied to blood, brain, liver, breast epithelium, or skin. No tissue-specific calibration required. One pan-tissue clock for the entire organism. The implication was significant. A universal methylation signature for age, functioning identically across every tissue type, implied the existence of a single biological aging process underlying all of them. Horvath described the clock as measuring what he termed the cumulative work of an epigenetic maintenance system, the cellular machinery responsible for preserving the switching architecture against the pressure of time and environmental exposure. The most striking result from that initial paper was not the clock's accuracy on normal tissue. It was what happened when the algorithm was applied to cancer. Across all 20 tumor types examined, cancer tissue showed a mean biological age acceleration of 36 years above matched normal tissue from the same patient. The molecular clock registered a biological debt in cancerous cells that had accumulated decades ahead of schedule. 2 years later, a prospective multi-cohort study published in Genome Biology by Marioni and colleagues demonstrated that the deviation of biological age from chronological age carried an independent mortality signal. In longitudinal cohorts of older adults, with a combined population exceeding 4,000 subjects, each 5-year increase in biological age acceleration was associated with approximately 21% higher all-cause mortality risk, a figure that remained at 16% after full covariate adjustment. The clock was not measuring time. It was tracking the accumulation of biological liability with measurable consequences for survival. But the first-generation clock, by design, was trained to answer one question: how old are your tissues? The more consequential question is what that age predicts about outcomes, and that is the question the next generation of clocks was built to answer. GrimAge, published in 2019 by Ak Lu and Steve Horvath, shifted the methodology. Instead of predicting how old your tissues appear, it was trained to predict mortality outcomes directly. Across 2,105 adults tracked for 17.5 years, each 5-year increase in GrimAge acceleration carried a hazard ratio of 1.50. A hazard ratio is a standard risk measure from clinical research. A ratio of 1.50 means 50% higher mortality probability against the baseline population. Individuals running approximately 8.3 years biologically older than their chronological age carried more than double the mortality hazard. Those running 7.5 years younger carried roughly half. Same birthday range, different biological trajectory. But position alone does not tell you velocity. Consider the difference between an odometer and a speedometer. The odometer measures accumulated distance from factory condition. The speedometer measures how fast the vehicle is currently moving. These are not the same instrument. They answer different questions. DunedinPACE, developed by Belsky at Duke University and published in eLife in 2022, is the speedometer equivalent in biological aging research. A score of 1.0 reflects average aging, 1 biological year of measurable deterioration per calendar year. A score of 1.2 indicates a 20% acceleration in that rate. Within the same research, a 12% average reduction in caloric intake measurably slowed that score over 2 years. The rate responds to measurable inputs, which means the clock is not passive, it is a potential target. The most consequential finding in this research trajectory is not a measurement. It is what happens when the measurement changes direction. The TRIIM trial, published in Aging Cell by Fahey and colleagues in 2019, enrolled 9 healthy men in a 12-month intervention. One system requires context first. Your immune system depends on a central production facility. Think of it as the manufacturing and quality assurance department of your body's defense infrastructure. It trains the workforce and maintains output standards. Early in life, it operates at full capacity. Beginning at puberty, it contracts. By the mid-60s, much of that original capacity has been replaced by inactive tissue. The clinical term is thymic involution. As the facility contracts, immune output and calibration degrade in step. The TRIIM protocol used recombinant human growth hormone, metformin, and DHEA, targeting partial restoration of that contracting facility. At 12 months, the Horvath clock showed a mean reduction of approximately 1.5 years from baseline. Against the biological age the subjects were expected to reach over the same period, the net figure approached 2.5 years. GrimAge showed a reduction that persisted at 18 months, 6 months after all treatment had stopped. The caveats are documented in the original paper. 9 subjects, no randomized control group, replication in larger populations penned in. Within those constraints, the result is this: a measurable reduction in biological age on peer-reviewed epigenetic clocks is an achievable experimental outcome in living human subjects. The clock, under these experimental conditions, is not read-only. Convergent evidence comes from Dornay and colleagues in a 2022 preprint, currently pending peer review. Individuals who reached 100 years of chronological age showed DNA methylation ages running 15 to 28 years below that figure. Their biological children showed methylation ages running 4 to 12 years below age-matched peers. 2 separate populations, same direction. Before this briefing, chronological age was the accepted proxy for biological state. Aging was a process medicine could observe and manage, but not quantify at the molecular level with precision sufficient to predict individual mortality outcomes. What the evidence establishes is a different model. The molecular switching system inside your cells, what the research literature calls the DNA methylation clock, predicted all-cause mortality in several large prospective studies more strongly than the standard aging markers currently in clinical use. That mortality signal has a rate. The rate diverges between individuals. It responds to measured inputs, and in at least one controlled human trial, it moved in the opposite direction. Your chronological age is a record of time elapsed. Your epigenetic age is a record of biological state. The research is now building a measurable, testable line between the two.